Okay, great. So again, hello from my side, and welcome to our webinar today. So I'm very happy that we have today Yolanda, or now Privatdozent Dr. Yolanda Salinas, here for a talk on Mesoporous Silica nanomotors as active drug delivery nanovehicles. And so to say a little bit about Yolanda, so she graduated as a chemical engineer in Valencia and has also done her PhD there. After that, she went to London with a Marie Curie Experience Researcher postdoc position. And finally, she ended up at Johannes Kepler University in Linz at the Institute of Polymer Chemistry, where she works a lot on mesoporous nanoparticles and mesoporous silica nanomotors and all these things, so very related to molecular communication. And I also had the honor to work with her a few years ago on some papers, on some projects, and I hope we can continue that soon. And I'm also very happy to have her here today for this talk. So Yolanda, I will stop now sharing my screen and you can share yours, and then you can directly start with your talk.
Thank you, Dr. Maximilian Schäfer. It's a honor to be here to explain my topic to the community. And I will share my screen now, so you can see the slides. So today I will talk about a very specific topic, that is nanovehicles made with mesoporous silica particles, and they are used for active drug delivery. So initially, I will introduce nanomaterials and nanomedicine, because I would like you to understand better now what is a nanoparticle. And we have to start thinking about how small is a nanoparticle. Well, if we think about it, a nanoparticle will be the size of a football, then a virus will be as big as a person, and then a donut will be as big as New Zealand, and a kiwi will be as big as the world. So you can imagine how tiny these nanoparticles are. Due to this very, very small size, they have enormous surface to volume weight ratio, which means that they have special properties that differ from the bulk material. And if we want to compare them in the diameters or the size of more biological molecules, we can make it here in this scale, where we can observe the size of, for example, glucose, protein, DNA, or viruses that we are more familiarized with. So nanoparticles will be in this range of a specific size, from 1 to 100 nanometers. But a key point in the successful of nanotechnology itself is the way that how we prepare these nanoparticles. So in the 1960s, there was the famous physicist Richard Feynman, who says there is plenty of room at the bottom, which means that there is a way to make in a small particles down a bulk material in a way that we physically grind or physically erode this bulk material and make it in the very tiny particles. But there was a contradictory talk from a very famous Nobel Prize in Chemistry, Jean-Marie Lehn, who says there is even more space or more room at the top, because the nanoparticles can be produced by chemical methods, by typically synthesis, by improving the synthesis and creating specifically linkers between atoms and becoming clusters, and then obtaining nanoparticles in a specific properties and characteristics. So we can think now about this nanotechnology to be in use in nanomedicine. And this is fascinating because it will create the future fundings of cures for many specific diseases. But nanotechnology, in this case, applied for medical and nanomedicine, can help us also to prevent diseases. So we are developing new techniques and new nanomaterials that could be achieved in a better way and obtain better results for theranostics and diagnostics. So we are ending in a new era of nanotechnology, where we are pursuing the targeting of, for example, repairing damaged structures, finding and treating diseases, deliver some specific drugs on specific target points like cancer cells or tumors, or even start making some bioengineering inside the cells. So this is very fancy and very futuristic, but the main targeting point here is to treat cancer and to deal with this, one of these leading causes of mortality nowadays, of course, followed by the cardiovascular diseases. So cancer is a very particular disease that is characterized by a very fast proliferation of growing of cells and very low cell death, which means that they will end up accumulating and forming like an abnormal mass and forming this kind of tumors that we unfortunately we know. So if fortunately this has been localized and we know where it is, so the medical doctors can treat this by surgery or by radiotherapy. However, there is a very fast vascularity production, and this ends up in very fast spreading of the cancer cells around the body, which means that they will end up in metastasis, which is one of the leading causes of mortality. So one of the traditional ways to treat cancer is by using chemotherapeutic drugs. However, the treatment of this to the patients is very brutal, I will say, because we introduce very big amounts of these chemotherapeutic drugs, which acts like a poison, let's say, which kills indiscriminately cancer cells and healthy cells, which also will end up making multi-drug resistance to the patient and ending up in very secondary effects like the patient feeling dizzy, feeling with nausea or vomiting, and losing the hair, for example. So the use of more specifically intelligent materials that could trap this drug and could be able to move this drug towards the specific target points, that will be the solution of these problematics that we find nowadays with the treatment of cancer. So here I wanted to show you these very also futuristic images, which shows how the nanotechnology at the end will be able, hopefully, to fronting personalized cancer by these specific nanomaterials or nanomotors, and even treat tumors or solid tumors and specifically localize them or even stop the reproducing of these malignancy cells before they made these tumors. So can we say that the nanotechnology is the future of medicine? Well, we are in this way, and we hope that this will be the way, because this will mean a lot for the cure of new diseases, like, for example, cancer. This is not new, but it is expected that the global market in this year will be exceeding $125 billion, and in the next year, even the possibility to create like a smart pill, it will increase also the commercial market. But this is very expectations that are actually not happening in nowadays so easily. So still we have to treat the nanoparticles very carefully. We have to develop them in a way that they are not creating other secondary effects. And in the scientific community, many people is working and publishing articles for treating, for example, different types of cancer. In this case, there are many publications for lung cancer, followed by pancreas and prostata, and then for ovary cancers. But in this case, this developing, it is moving toward three types of approaches. First one will be drug delivery, like we can make some therapy to the patient. The second one will be imaging, so we can detect, we can find where the tumor is located. And the last one will be theranostics, which that there is a therapy and diagnosis. So we are able to aim both. So as you can see, there is an evolution on this type of drug delivery systems since the middle of the 20s. So actually, on the 20th century, actually there was at the beginning of the 50s, that there was a creation of these typical tablets, capsules. This was the common way to introduce drugs as anti-cancer drugs, or for example, any type of drugs for treating diseases. That in this case they were opening the capsule and releasing the drug in a burst way. So there was not a sustained control. There was not a control under the time of the release of this drug. So as we can see, after a few years, the scientists developed this type of more sustained release. Later on, more different type of materials were appearing, like, for example, liposomes or functional carriers, which carried some specific target units, which could detect specifically the cancer cells, and so on and so on. So there were many different types of materials that are being created in order to be used as drug delivery systems. But nowadays, we are ending in an era that the more active drug release is needed, and by this way, in the last 10 years has been a very good development of micro and nanomotors. Nowadays also we are ending in a more sustainable set of mind. So we have to create more degradable materials that, apart that not affecting the body when we intake it, that they are not affecting the ecosystem or the environment. So when the nanoparticles are delivered in the system and they are encountering the systematic administration in 3 different phases. So they pass from the phase 1, that is circulation. So the nanoparticles are circulating, are moving around the blood vessels. And due to very enhanced permeability, retention of the tumors, then the particles can be accumulated in the tumors. And afterwards, they will be internalized inside the cancer cells, and then it will be the drug delivery happening. As you can see here, after the passing the phase 2, that is the most dramatic one, we have the accumulation and the penetration of these materials through the cancer cells or tumor walls. Once they are accumulated in the surroundings, they can entering due to some receptor-mediated endocytosis, and then afterwards by forming these endo and lysosomal structures, they can be able to release some drugs specifically due to the specific pathological conditions. So in order to develop these materials, we can play around with different type of chemistries, let's say. So we can use, for example, more organic materials, and we create organic nanomedicines. In this case, we use more polymeric systems. For example, drug polymer conjugated. So we have a polymer with a drug attached on the side, and we can create also micelles, or we are able to create some liposomes where the drug is inside the liposome, or polymeric nanoparticles. Also, the drug is incorporated inside the polymeric nanoparticle. Dendrimers, where the drug is attached in one of their branches or in the core of this dendrimer, and protein polymer conjugates. So these are specifically more organic nanomedicines. But we also have inorganic nanomedicines that are very interesting. For example, we have silica nanoparticles, which I will talk to you about later. We have hollow porous nanoparticles. We have also gold nanoparticles for imaging. We have iron oxide nanoparticles, quantum dots, or carbon nanotubes. So these are very among the most used inorganic nanomedicines. So we can see that there is a strong bonding between properties when we combine organic and inorganic. So it is possible to use the specific materials properties of polymers, like, for example, easy processing, lightweight, flexibility, and very versatile structural and architectural characteristics, combined with inorganic materials that they present more superior thermal and mechanical behavior, and also optical, electrical, catalytic, and magnetic properties. So we will create a hybrid system by combination of organic and inorganic units. So it is kind of like playing with Lego. So we combine units, in this case, different components together, and we create more complex system from a more simple system or blocks. So Now I would like to pass to the second topic, that is my main research based on silica nanomaterials for being used in nanomedicine. We can create from silica nanoparticles different size and different shapes. So we can create from very tiny nanoparticles to bigger ones, from nanoparticles to microparticles. We can also change the shape to more spherical, to more rod-like structures or nanorod nanotubes. And very interestingly, we can modify the surface of this type of nanoparticles by changing the molecules that are in the surrounding of them. For example, attaching some negative or positively charged units or polymers, and then, for example, make them more hydrophilic or more hydrophobic. We can also incorporate polymers that will contain target ligands, which means that they could be very useful for targeting purposes. So, as you can see, silica nanomaterials can be developed as a non-porous, like we have here, and these non-porous silica nanoparticles, they have very shell structure. Sorry, they have very shell structure that are containing this cargo, and they can be releasing this cargo by degradation. Also, we can incorporate the cargo, that is the anti-cancer drug, by attaching it to the surface. So this is the structure that really, in real, how they look like. So they are spherical and very homogeneous. But we are able to also obtain materials that contains porous. So this is called porous silica nanoparticles. And this is the very interesting ones for me, and this is my main topic. It's called Mesoporous Silica nanoparticle, MSN. They are containing very longitudinal and very non-connected tubes that can be uploaded with drugs, but they can be also kept with some specific molecules, which we call gatekeepers. These molecules could be from polymers to organic, other organic units, or more inorganic, like nanoparticles, for example. So this will prevent the release of the drug under demand. And this material, it looks like this under the transmission electron microscope. So it is very porous and very homogeneous in morphology. So what we are creating here is kind of like a taxi. This material, it is transporting the drug, as you can see here. We put here the drug in the truck, and it is transporting the drug through the blood vessel. When the material or the nanoparticle has reached the point or the target, the traffic light is on. So it means that the gate can be open and the release will happen. So these materials are very controllable in terms of on-off drug delivery. And as well, these mesoporous silica nanoparticles, they are biocompatible, they are non-toxic. They can have a very high loading of drugs. They are able to avoid this zero premature release. They have a very efficient cellular uptake, and they have been created in a way that they can be controlled the release among a specific stimulus. So my main target in research, it was to do materials that can be degradable. So these mesoporous silica nanoparticles will be degraded under specific conditions and time. The second one is to control better the release of the drug by using specifically more degradable gatekeepers, like in this case, polyphosphacins. And the last one, it is the use of these kind of polymers to create a more motor-driven system that could be enclosing one of the side and leaving the other side free. So the release could happen and move forward the nanoparticle. Based on that, my first topic is the use of these materials for degradability. So what I am trying here is to develop a system that in this case you can see here is a mesoporous particle that after being containing the drug and transporting the drug in the blood, normal vascularity vessels, they can be collected here and accumulated in the cancer cells or tumors, and only under specific conditions, pathologically conditions like pH, temperature, and so on, they will be able to release the drug. And afterwards, these nanoparticles will not be staying in the body and accumulating, who knows, somewhere in some organs, but they will be able to degrade after being able to release the drug. For that, we select polyorganophosphacins because these polymers are very tailored materials that can be prepared or synthesized in a way that they can be degrading under specific conditions and form small molecules or phosphates and ammonia, which are harmless products. So this type of polymer, it is containing phosphorus and nitrogen in the backbone and chlorine atoms attached to the phosphor atom. So by macromolecular substitution, we are able to create a hybrid containing organic units here that could modify the properties of the polymer and also make it specifically degradable under specific conditions. So, but for being used for our nanoparticles, our silica particles need to have a specific alkoxysilane linkers. So in this case, we derivatize the polymer and we attach alkoxysilane units on one of these phosphorus atoms. Based on this specific objective, we have the particle that contain the silica units and also the phosphacin units, which are degradable. Therefore, under specific conditions, there will be a possibility that these phosphorus atoms or phosphor linkers, they will start degrading, and this polymer start helping to the disassembling of the nanoparticle. So the particle will break up quickly. As we can see, this was one of the promising results we obtained, that after 1 week at pH 2, the nanoparticles start actually degrading or decomposing. In a second target aim was like to create the porosity that we were expecting, because we want to have this hexagonal, very nice arrangement of pores. But somehow we were not able to obtain this porosity, but different type of porosity in this case, like more like a foam. So this foam also contain particle. This contains specific cavities that could be uploaded or linked with some specific cancer drugs. But that was very promising and proof of concept, yet not conclusive, because it allowed us to observe that there was the possibility, because for the first time we used together polyphosphacins and silica. So this material was developed by my PhD student, Vanessa Poscher. And after many, many trials, we observed that the material was able to degrade even under pH 7 after 4 weeks. But based on my experience and the things that I was learning during my doctorate studies under the supervision of Professor Ramón Martínez Mañez at the University Polytechnical of Valencia, we were able to create some supramolecular nanoarchitectures. These ones contain 3 units. The first one is the Mesoporous Silica support. The second one is the cargo. And the last one is the functional molecules, polymers, inorganic nanoparticles, and so on. So after a specific external stimuli, like light, heat, or magnetic field, or internal stimuli, like lysosomes, enzymes, or different pH or redox conditions, this material on top, it was able to change the conformation and release the drug. So here we can see how these polymeric gates were used for controlling the release of the cargo. So once they are covering the particle, these kind of hairs are not allowing the cargo to be released. When the polymer is collapsed, we can observe that the release happen upon this stimulus. In this case, we create a polymer that was able to have thermoresponsiveness. So we can see here that the polymer in water, it was transparent and it was all dissolved. But when the temperature increased, we can see the aggregation of this polymer and forming this cloudy solution observable by naked eye. So that was something that I also studied during my postdoc in Queen Mary University of London, where we developed specific nanogels cross-linked to be able to use for drug delivery in skin cancer. So in this case, the material will contain, in this case, water molecules or drugs, and it could release this depending on the temperature. So we can see that there was some kind of thermoresponsiveness very nicely after several degrees of differences. So based on that typical nanogel, we use these polymers like polyphosphacins to create a more bottle brush type, which could be collapsed as well and let the release to happen. Here I am showing you that there is a possibility to increase or decrease the hydrophilicity of this material just by changing the organic sites. So we use this polymer and we attach it to the silica particles, and we could see that below the LCST, which is the thermoresponsiveness of this polymer, there was almost no release happening, as we can see here. But when the pH was below neutral pH, we could see a very increase and release of the drug. So this material was sent to Barcelona to my collaborators in the Universitat Ramon Llull, and they were able to see how these particles were accumulated inside the cancer cells and release the drug after a while. In this case, we use camptothecin as a specific chemotherapeutic drug. So we are observing that there is a road ahead in the future of drug delivery systems. So starting from the traditional carriers to the more targeted carriers and micro devices. But in microrobotics, there was also an evolution between a silence period from the time where Isaac Asimov described in his book, The Fantastic Voyage, the use of nanomotors or nanoparticles that could be kind of like autos or cars coming to the blood vessels, to more outbreaking period and the future of the microrobotics. So these particles or microrobots will be able to navigate through the blood, will be able to target tumors, to penetrate in tumors. They will be able to also be uptake cellularly, release the drug, and even do delivery in gastrointestinal problems. But what is a micromotor or nanomotor? Well, it is a particle that can perform a motion, an autonomous motion or movement in a complex fluid driven by different power sources. So in this case, the possibility to develop a Janus particle, which comes from the Greek god Janus, which had two faces, we were able to make nanoparticles with two different faces in different ways. But in terms of the scientific developments for mesoporous silica nanoparticles, there were not many, or kind of, there was a very minimum amount of papers where these particles were becoming nanomotors. And in a way to develop them, we had to check which kind of common way of propellation the material could move. So we can create chemical propel systems by using hydrogen peroxide, which will release oxygen bubbles and will move the particles towards. Or they can be also created by fuel free, where magnetic field, near infrared light, or any other source, external source, will be able to move these particles forward. Or even created by biomimicking other biomolecules to obtain biohybrids. In this case, my interest was in preparing Janus particles, which has chemically propelled motion. So the first trial was to put the polymer around and see if we were able to control the switching on and off of this releasing of the bubbles. We use different type of catalyst inside the pores, and we were able to see the degradation of this hydrogen peroxide, that is the chemical fuel, into water and oxygen bubbles. And this gas was able to be controlled like on-off by changing the temperature, as we can see here. So between 15 to 20 degrees, we saw almost no bubbles happening. 25 degrees, the bubbles start forming. Then we see 30 degrees, more bubbles. And when we decrease the temperature, this stops. And we could do switch on, switch off for several times, which was very, very interesting. But our main aim was to make this power nanomaterial to be moved forward. So therefore, we create only functionalization side of the particles. So this Janus grafted micromotor will be able then to move in a one direction, because there will be only the entrance allowance for one of the sides. And here I want to show you very nicely how the promising results we obtain, where we can see how these motors are really moving around in the aqueous suspension under the microscope. We use just an optical microscope that we could see that. I will press it again because it's very nice. And I will explain you also that after being able to switch on and switch off the thermoresponsiveness, we can also make these nanomotors to stop. So these were the Janus particles, which at 20 degrees they stop, the speed is reduced dramatically. And when they were fully grafted, there was no this transition happening. So that was very interesting, not only for the scientific community, but also for the public, because we were advertised in one of the newspapers from Austria, very famous, Der Standard, and they published this article about us, about the results that we obtained, like nanomachines obtaining some flexible brakes. We also got a front cover in the Chemistry European Journal, which we were very proud of. And I wanted to show you here one of the cartoons that I designed together with Animating Creating Research Wiki. And they developed this cartoon for us, which it is representing submarine, that is our nanomotor, that is degrading after opening the gates and releasing the drag. And now I'm going to talk about the last, maybe more interesting topic for you, but this is not my expertise, so I will speak about this briefly. So there will be possibility to combine the knowledge of telecommunications and also the chemistry behind nanomotors. So there is a potential synergy. We observed that already from the publication from my former supervisor, Ramon Martinez, who published the possibility to apply an external input, which end up with sending a messenger out of the particle, of one of the particles. This messenger will give another input and then output coming from this particle. And then finally, the last output will be released. Based on that, they developed one of the first examples on Mesoporous Silica nanoparticles communication, where specifically particles designed like in a snowball structure we have the mesoporous particle, and we have a gold nanoparticle attached with some enzymes. So the use of input like lactose will produce an output like glucose that later on produce some make a reaction chain forming gluconic acid in contact with this enzyme, decreasing the pH, and then opening one of the gates with this cyclodextrin, and then releasing the drug or some specific suitable dye that induce the release of another suitable dye in the last particle, in the first particle. So based on that, there is kind of a communication between these nanoparticles, though they talk to each other. And there is an interesting possibility to create communication by using this molecular communication experience that you have, guys. So, for example, encoding some messages inside the nanoparticles that they will release this information and they will propagate through the specific medium and then end up in receivers that will decode this information afterwards. So based on that, we create some kind of channel modeling application where we can study better between our collaboration with Dr. Maximilian Schäfer and Professor Werner Hasselmeier. We can understand better mathematically and modeling what is happening with the release between the matrix-based nanoparticle and the absorbing receiver. So there is initial calculations, mathematical modelings, that will allow us to be able to design in a better way our nanoparticles to obtain more improved systems. So I have to say there is a very promising future for nanomedicine in many different topics like diagnostic, medical imaging, nanotherapeutics, regenerative medicine, and vaccines. So there is a very huge future in front of us that with the putting together all our backgrounds and minds, we can develop very good things for the future. So I want to acknowledge my supervisor for the postdoc, Professor Oliver Brueggemann, also my collaborator Jan Tisdel, and my students, of course, for all the efforts done in the lab, and the whole team for the support. And especially acknowledgements to my collaborators, as you can see here in the list. And definitely thank you all for all your attention. So I am happy to reply to your questions, comments, or concerns. Thank you so much.
So thank you very much, Yolanda, for the nice talk. So are there any questions from the audience? Just unmute yourself and then Yolanda is happy to answer your questions.
Don't be shy. No questions. I'm sorry if my voice was a bit timbling. I was under the weather.
So then maybe I can start, Yolanda.
Definitely.
So I knew some parts of the talk already.
Mm-hmm.
So but for the motors, I have the question. So like let's say in a medical application, what is the big benefit of having these nanoparticles moving basically faster around compared to having no motor? What is the
That's it.
benefit in this sense?
Yeah, that's a very good question. So actually, the fact that they can move, they can be improve the release. They can target better the tumor because sometimes the particles are moved throughout the whole blood vessels from the time that you inject it until they get out of the body, of the human body. So in this case, if they have some kind of propulsion, they are maybe able to accumulate better and further through the tumor also walls, because sometimes we just expect that they will be endositized and they will be capped, but sometimes they just pass and they just don't touch even the tumor. So in a way that they will be more faster activating the drug release, I will say. But this is very big concern for this community working on the nanomaterials.
Which then is a big concern.
The how is this gonna work in the human body? Like what is gonna happen? Because these are all hypotheses, but we don't know um, what is gonna happen in real inside the body. So we can simulate this in the laboratory. We can make plans. We can make theories about it, but until we try it in, for example, animal models or even in the humans, we are not able to see exactly how it's gonna be this more beneficial. Let's say we have, of course, the background that it is gonna help, and we can see in the results I show you that the particles are accumulated in the cancer cells, not in the healthy cells. So they are working, and it is happening that these are very potential results for not from me, but in the whole scientific community, that they can be applied for curing or treating the cancer. And the use of nanomotors is kind of still new, it's still in developing. So just for the last, let's say, 15 years and the last 10 years, there were more evolving and more designs happening and being made by scientifics on this topic. So there is a lot of hypotheses behind, yeah.
Okay, thank you.
Thank you.
Any other questions? So maybe Muhammad was first.
I guess you open the floor, eh?
Yeah. Thank you, Yolanda, for giving such an insightful talk.
Thank you.
We really appreciate it. So my name is Usman. I'm working with Professor Lin in Tongji University, China. My question to you is actually there are a lot of questions that I want you to answer, but I choose one. My question to you is, as you mentioned in your talk, that from degradation of nanoparticles, we need specific condition in the specific area where tumor is located. So what are the how we can make sure there is a special or specific or appropriate condition near the tumor so nanoparticles can be degraded or can be reached at the special destination.
Okay, now thank you so much for your interest and for the question. I mean, when we design these nanomaterials, we think about ahead what are the conditions of the cancer cells. So we are designing them in advance, thinking which properties these polymers or nanoparticles should have in order to be degraded. So the idea is that in this case, in my materials, when they are releasing the drugs, then there is more contact also with the conditions from this tumor site. In this case, we talk about more acidic pH, higher temperatures. So the more acidic pH is the one we are using for this material to degrade. So it will end up cleaving one of the organic units linked to the polymers, and this will kind of make a chain reaction that the material will start degrading. And that's why I show you these particles degrading under pH conditions. So I hope I replied to your question. So we are just thinking ahead which conditions the tumor could have, so then we can create the material accordingly. This is the way, yeah.
Oh, thank you so much. And one last question, as you mentioned, nanoparticles. So we are actually from the molecular communication and particularly from the communication perspective. So I've seen a car in your slides that contains nanoparticles. And that car has the potential to release wherever we want. My question to you is, can we view that car as a transceiver, or because I just wanted to know what are the functions that can be performed using that car. like that car can sense surrounding, can release. So we can we view it as a transceiver?
I will say this is like the transmitter, but maybe Max can reply better to this because he's our collaborator. So this will be the transmitter system, and the receiver will be the tumor, if it's how you put it in this way. So there is the way that the this happening is the releasing of the drug modulation. You know what I mean? I mean, this is kind of uh, putting in a box, let's say this motor, and then after releasing the drug, we see that these molecules are reaching the receptor, that is the tumor. So that will
Okay.
be the way to make mathematical modeling. But as I say, these are a part of my collaborators, so I cannot explain much better this topic. Yeah.
Okay, thank you so much.
Thank you. Thank you.
Thank you.
Sure, Daddy, does go on. Right.
Hey,
Hello.
Yolanda, thanks for a nice talk. Yeah, I
Thank
have
you.
a question about the motor you designed. So, as mentioned in the slides, it seems your motor only has two states, on and off. So I'm a bit curious, is there a way to precisely control the direction of the moment? For example, can we tell the motor, okay, next step, let's turn left and or turn left or turn right and
Yes, yes.
or backwards? Yeah.
Yeah, thank you, thank you. That's a really nice question, actually, and this is the next step on my research because I also thought about it. Now we just cover one side with one polymer, so it could move in that way, let's say in one direction. But if you put in the other side a different material or different responsive system, then you could control which way the particle will move in left side or right side, or so it depends on the switching, on the stimulus you use. You can make the material to move in one direction or the other direction. That will be the future of this. Yeah.
Okay.
Thank you so much. Very, very nice.
Thank you. Yeah, thank you.
Okay. Hello, Yolanda. Can you hear me
Hello.
now?
Yes, we can hear you.
Okay, first, thank you for your nice presentation. I have a question about the trade-off between cytotoxicity and the efficacy of the operation, because I haven't heard, and as far as I know, in this sort of drug delivery, while coating the drug with nanomaterial, one of the main concerns are the cytotoxicity that actually appears in the system. So I wanted to know, in your series of works, have you addressed this trade-off between the two? How did you define the efficiency and the cytotoxicity? Were there any trade-off between the two?
Thank you so much. If I understood well, you mean if both units, the polymers and the silica particles, are both not toxic, let's say? Is this a question? Yes, we do this as well because, for example, the silica particles, they are well known not to be toxic. They are biocompatible. And on top, this polymer, it has been developed in my institute for a long while. And apart from changing the properties with the organic units, we choose the organic units that are not toxic. So everything we use should be not affecting negatively. So we try the cytotoxicity and we test it, and it was no toxicity shown. Yeah.
So regardless of the amount of nanoparticle you use, the system behavior would be the same.
Yes. Yeah. I mean, of course, we tried with a specific test experiment, like if I remember well, it was 100 milligrams, 50, and so on. So we go from like the common test. So my collaborators did that in Barcelona, and they do the typical experiment. And we didn't try 1 kilo of particles, but with a small amount, we can see that there is not affecting negatively to the cells and they are still alive. Yeah.
Yes,
Thank you.
thank you.
Okay. Do we have further questions from anyone? Okay, seems to be not the case. So then, thank you very much again, Yolanda. Thank you all for attending the seminar.
Thank
So
you.
you will find the video on YouTube soon. And I will also send an email around, or we will send an email around soon about the next seminar we are planning. So thank you for being here.
Thank you so much. Bye-bye.